All right. We ready to rock and roll? No? Yeah, it's too early, right? Me too. I don't envy you guys. Every day at 8. My brain doesn't work very well at 8, as you guys probably figured out, right? Um, okay, so today we're going to talk about the pentose phosphate pathway. Actually, we're going to talk about a couple things today. That's the name of the lecture. We're also going to talk a little bit about ethanol metabolism. Dr. Traore and I discussed we needed to squeeze in ethanol metabolism somewhere, and this was a shorter lecture, so we're going to do it at the end of that, at the end of this lecture. But it actually, with how we're approaching the subjects, the ethanol metabolism sort of fits in nicely because you guys just covered glycolysis and gluconeogenesis, so we'll talk a little bit about ethanol metabolism as well. But the first thing we're going to talk about is the pentose phosphate pathway. I call it the HMP pathway, which is the hexomonophosphate pathway. You also see it called the pentose shunt pathway, hexoshunt pathway. So they'll use that term shunt a lot because it is a shunt off of glycolysis, as we're going to see. So it's a branch off the glycolytic pathway, and it serves two purposes. And I'd like you to know what the two purposes are. Just like with how I approach other biosynthetic pathways, first start with the big picture. What are we doing here? Why are we doing it? And then focus in on the key enzymes and how they're regulated, and you know the pathway. Okay? And you know it in as much detail as you ever need to know. So this pathway's got two purposes. One is the generation of NADPH. Okay, this is a little bit different than NADH. NADPH, and I'm going to show you some of the things we use NADPH for. It's a similar idea, though. The NADPH is the reduced form. It's the high energy form, and we can use it for different things, including synthesis of certain molecules. And the other purpose of this pathway is to generate a 5-carbon sugar ribose, which is something you've seen before, right? You've seen ribose and you've seen deoxyribose. We use those to make nucleotides, okay? And then we use nucleotides to make nucleic acids, and we also use nucleotides as a source of energy. So this is the pathway where we actually synthesize the sugar that will be part of the nucleotide structure, okay? So the pathway can produce both ribose and NADPH. It can produce both of them, or it can only produce one or the other if you would like. And actually, that'll be even more clear on this next figure. So we can come in and we can go through the process of producing NADPH and then go into the process of producing a 5-carbon sugar, so we can actually do both. Or we can produce NADPH and then we can actually go back this way and go into, what's this pathway? Starting with glucose and ending with pyruvate. That's glycolysis, right? So you've already seen that. So you can go in, make NADPH. If you don't need 5-carbon sugars, you can shunt it back in, back into glycolysis. Or if you don't need NADPH, but you do need ribose, you do need nucleotides, you could actually skip this step and actually go here. So the arrows are important in terms of the directions here. Now, this is a figure I took from a Comlex review book, actually USMLE review book. Either way, it's the Kaplan review book. So you guys will get that book eventually. I think they've actually even updated the figure now, and it's the same figure, it's just in color now. I like this figure because it cut out a lot of extra information, so therefore I'm going to use it in this lecture. The one in your textbook is much prettier, and but it's also a little more busy. The thing I really like about this particular figure is it cut out all the enzymes except the two that I want you to know. Okay, and the two key enzymes, and I'm going to go over those and why I want you to know them. Okay, so that's the purpose of the pathway. So there's two. There's two types of reactions in this pathway. And And the first, and there's so basically two phases. The first is this irreversible oxidative phase that generates NADPH. Okay, so that's one of the purposes of this pathway, right? We're going to generate NADPH. So the major enzyme, and this is the rate-limiting enzyme, this is the key regulatory enzyme for this pathway. Remember, whenever you hear that, that's how we're going to control a pathway. So this is the big one. This enzyme is glucose-6-phosphate dehydrogenase. Okay, big long name. Do you need to know it? Absolutely. You're going to see there's a major, very common genetic disease associated with defect in this enzyme. You also see very frequently, and as I get later on, when I start talking about the deficiency of this enzyme, I'm going to use this abbreviation G6PD. It's much quicker to say G6PD, and maybe some of you have already, depending on how much you've studied this stuff before, have already seen this. G6PD is an abbreviation for glucose-6-phosphate dehydrogenase. So you should know both of those. Be comfortable with both of those in case you see them. The reaction is inhibited by high levels of NADPH. So if you build up NADPH, that's substrate inhibition, or I'm sorry, product inhibition, right? So that kind of makes sense. Insulin actually increases transcription of the gene for G6PD. So what does that tell us? What does insulin indicate? Well-fed state, right? You're taking in extra whatever, carbs, other things. So that means you're in synthesis mode. You're going to make things, right? Well, NADPH is going to be used to make things. Fats, for instance, cholesterol, for instance. Actually, even nucleotides themselves use NADPH. And also, you know, if you're in a mode where you're getting lots of fuel, you may want to actually, undergo cell division. So you may need to make nucleotides, okay? So that makes sense. So this would be a pathway that's going to be most active in the well-fed state. And you'll also notice this is the third pathway that you guys have seen for glucose. Once it enters the cell, it becomes glucose-6-phosphate. We already talked about that. I believe yesterday you saw that you can actually convert that into glucose-1-phosphate and go into glycogen synthesis, right? But here's another place you can go. So these are two alternatives that you can go besides glycolysis itself. And then we also have a reversible, so you can see we got the arrows going in both directions, non-oxidative phase that can generate ribose. Now, as you're going through this, there's a little bit of extra information in there, and I don't want you to worry about that. Like, they have some of the intermediate sugars in there. Don't try to memorize those. I want you to know these two key enzymes. I want you to know we're making NADPH and we're making ribose. Don't make it any more complicated than that. And realize, you know, this is an alternative place for glucose to go. Glucose-6-phosphate, you know, you have glycolysis, you can go into glycogen synthesis, or you can go into HMP pathway. Okay, so the extra stuff is peripheral information just to be complete. And once again, also notice that we can make NADPH, ribose, and go into nucleotide synthesis, or we can make NADPH and then actually convert that ribose back into intermediates for glycolysis, or we could skip making NADPH and actually make ribose. So you can do either or. You can do both or either or, okay? Not too bad, right? Okay, so there is one extra enzyme. So glucose-6-phosphate dehydrogenase is the key regulatory enzyme that I want you to know. And we're going to talk about that more later on in this lecture. But there's one other enzyme in there I told you need to know, and it's transketolase. Now, the enzymatic reaction that transketolase catalyzes is what a medical biochemist would call kind of boring, because it goes in both directions. So it's really not a key regulatory enzyme, so it normally wouldn't be something we would spend a lot of time talking about. However, transketolase actually has a thiamine requirement, Okay, that TPP right there is thiamine pyrophosphate. That's the active form of thiamine. Okay, so that's what that TPP means in this figure. Okay, so this is the fourth and final enzyme in the body that requires thiamine, and you've seen the other three, right? Pyruvate dehydrogenase complex, which branch glycolysis to TCA cycle, right? And then alpha-ketoglutarate dehydrogenase complex, which is part of the TCA cycle. Remember where the third one we saw was? That branch chain alpha-keto acid dehydrogenase that was defective in maple syrup urine disease, that had a thiamine requirement. And here's the fourth one. And this is arguably the most boring one because it's not really a special pathway in particular. But this is the one thiamine requiring enzyme that is actually still present in red blood cells. So this is the enzyme they'll actually use to try to diagnose a thiamine deficiency. Okay? So a thiamine deficiency usually doesn't have a direct effect on red blood cells per se, so it's not that big a deal if the transketolase is not working in the red blood cells up to 100%. But you can test for it. The other three enzymes that require thiamine are a much bigger deal. Okay? But because it's much easier to get a sample of blood cells than other tissues, right, this is something very convenient to check for. So what they'll do is they'll draw a blood sample, and they'll actually test both with and without thiamine. So they'll basically test for transketolase activity. If adding exogenous thiamine actually makes it go faster, that means you were deficient to begin with. Okay? So this is how they actually try to diagnose a thiamine deficiency, even though this isn't the most critical enzyme that thiamine is required for. And once again, it's the only thiamine requiring enzyme that's going to be present in red blood cells. Remember, red blood cells, once they're mature, they don't have a nucleus anymore, so they're not going to make a whole bunch of things. And the biosynthetic pathways that are available in the red blood cells have been severely restricted as well. You know, you don't have mitochondria, for instance. So, okay, so one of the jobs of the HMP pathway, so the other job of the HMP pathway was make ribose, and you already know what you're going to do with ribose, right? You're going to make nucleotides. So the other thing we're going to do is make NADPH. You've already seen places where NADH is used, right? You generate NADH and you send it through the electron transport chain, or some reactions will use NADH, but I don't think you've seen NADPH yet. So there's 5 general things that we can use NADPH for, or this is a list of 5 of the possible things. 2 of them we're going to look at, and both of them involve reactive oxygen species. You do use NADPH for reductive biosynthesis, so you're going to see fatty acid synthesis. You're going to see cholesterol synthesis. At some point, I don't know if in biochemistry, but somewhere you will see steroid synthesis. You will see nucleotide synthesis. So there's different synthetic pathways that will use NADPH. So when you see those, just remember, oh yeah, the NADPH came from the HMP pathway. Now I should say there is other sources of NADPH, but this is the major one. It's also used for neutralization of reactive oxygen species, which we're going to look at in 1 second. The cytochrome P450 monooxygenase system. So this is something that you guys are going to run across when you start talking about metabolism of drugs for certain. You may see it in some other places as well. For example, biosynthesis of steroids. But they actually use NADPH in the process. Okay. For generation of oxygen-free radicals, we're going to talk about that in more detail in a couple slides here. And the synthesis of nitric oxide. So you'll notice 2 of them I bolded and underlined. Those are the 2 that I'm going to want you to know a little bit more about, which we're going to talk about. The other 3 you're going to encounter at some point later on in your studying. So when you are going through this PowerPoint, focus in on these 2. And you notice they're actually kind of the opposites. In one case, we're going to actually neutralize reactive oxygen species. In another case, we're actually going to purposely make reactive oxygen species. And I don't know, have you guys been introduced to reactive oxygen species at all yet? Yes? In here, I mean? In med school? Not. Okay, so you have talked about them. So basically, a reactive oxygen species, non-specific reactive compound, it's going to destroy things, right? It's going to react with proteins. It's going to react with fats. You get lipid peroxidation. So it's basically a non-specific destructive thing that's going to destroy anything it comes in contact with, right? So under most circumstances, reactive oxygen species would be a bad thing. You actually get reactive oxygen species. I don't know if I mentioned it in detail, but when we in the electron transport chain, it's a side effect of how the electron transport chain works. You get reactive oxygen species. So anyway, you have to have a way of dealing with this, or else, if you have no way of dealing with reactive oxygen species, eventually it's going to damage nucleic acids, cause mutations, and then could potentially cause cancer. But it certainly destroy things. Catalase, superoxide dismutase, and the glutathione system are three of the ways you can actually deal with reactive oxygen species. And the glutathione system is what we're going to focus in on, but you may have already seen catalase and superoxide dismutase in some other classes, or maybe even this class in some other lectures. I'm not going to talk about those today. So glutathione is the big one we're going to talk about today. There's a couple enzymes associated with this glutathione system, glutathione reductase and the glutathione peroxidase. I don't think it's important for you to try to memorize those two enzymes, nor catalase and superoxide dismutase, unless you've already seen them in another class. So just focus on remembering what we use glutathione for. Recognize glutathione, okay? So, what we can see here in this figure, let me make sure I've got it on, okay. So, in order to produce the reduced glutathione, the reduced form of glutathione is what you will actually use to neutralize reactive oxygen species. So, glutathione can exist in either a reduced or an oxidized state. So, glutathione, it's actually just this little 3 amino acid molecule. And one of the amino acids is cysteine. And if you remember the R groups of cysteine, what did cysteine have? Had a sulfhydryl group, right? So that sulfhydryl group is the critical component of glutathione. So that sulfhydryl group, when it exists as a sulfhydryl group, is reduced. And it can actually neutralize reactive oxygen species. In the process, it will actually become oxidized. And what happens if you take 2 cysteine residues and perform an oxidation? Get that disulfide bond, right? So basically, that's what's happening here. You're going to take 2 molecules of glutathione. You're going to use them in their reduced state. You're going to use those to neutralize reactive oxygen species. In this case, hydrogen peroxide. That's one example of a reactive oxygen species. And we're actually going to reduce it to water. Okay, and I think I just said all that. So you see what's going on there? So that's one of our systems, glutathione. And we require the reduced form. In order to make the reduced form, you need NADPH. Okay? And it works by, once it's in the reduced form, it reacts with a hydrogen peroxide molecule in this case. It actually reduces that to water, and in the process, it becomes oxidized. And then in order to put it back in the reduced state again so it can be used again, you actually need NADPH. Okay? So it's not super complicated, but what do you think is going to happen? Let's just ask, what do you think is going to happen if you're deficient in NADPH with this system? You're going to compromise your ability to what? Neutralize reactive oxygen species, right? Okay, so keep that in mind because we're going to talk about a disease in a couple of minutes where that actually happens. Okay. The other thing I said we were going to spend a little bit of time talking about was the generation of oxygen, reactive oxygen species, or oxygen-free radicals. Now, I just told you and went over why these are so bad. Reactive oxygen species react with everything. Anything they come in contact with, they kind of destroy. So why the heck would you ever want to make reactive oxygen species on purpose? Well, certainly you encounter things you would want to destroy, right? And so neutrophils and macrophages, I'm not going to spend a great deal of time talking about those. You've probably already been introduced to those in micro and immuno, right? You've already seen these. So these are special cell types that are going to fight off infection. They have a system which actually will generate reactive oxygen species or oxygen-free radicals to try to kill bacteria, for instance. One of the enzymes in there, now there's two enzymes, and you'll notice one's bold and one's underlined. That's the one that I want you to know because there's a disease associated with it. There's a couple enzymes, NADPH oxidase and myeloperoxidase, that are actually used to generate these free radicals. And the important thing with the biosynthetic pathway we're talking about right now is they require NADPH to do so. Okay? So this is another place where NADPH is used. Now, if you have a deficiency of NADPH oxidase, then these cell types don't have the ability to generate the oxygen-free radicals, fight infection. So without looking at the next slide, what do you predict would happen if you have a deficiency of this enzyme? It would compromise your ability to what? Fight infection, yeah. So that's what it's going to do. And there is a genetic disease where you can have a deficiency of this enzyme. It's called chronic granulomatous disease. And the symptoms, as you might imagine, are persistent severe infection. So bacteria that may not normally be known to cause severe infections in humans, because usually you can fight them off, may cause infections in these individuals. So basically you've compromised your ability to fight the infections. Now, you guys know a lot. You guys probably already know more about immuno than I do, but you realize there's more to the immune system than just that. So your entire ability to fight infection hasn't been compromised, but you've compromised it to a certain extent. One of the features of this disease, or one of the symptoms you will see in this disease, as the name implies, is the formation of granulomas. Now, granulomas can be formed in multiple different ways or for multiple different reasons, but it's always a walling off of something. But in this case, basically what happens is the macrophages, neutrophils, they're trying to fight the infection, and they're not, the bacteria are still persisting, so what the body does is just kind of wall it off. And when it does, it forms this granuloma. Okay, so you see both granulomas and persistent severe infections. So I'd like you to add that enzyme to the list of enzymes to know from this lecture. It's not going to be a huge list. I think that's the third one I asked you to know. Okay, so the next disease we're going to talk about is glucose 6-phosphate dehydrogenase deficiency. Much easier to say G6PD deficiency. Remember, what was this enzyme? This was the key rate-limiting enzyme for the HMP pathway, right? The gene for this particular enzyme is located on the X chromosome. What that means, and I know we haven't done genetics yet, that's coming up shortly, so you have to trust me here for now if you're not already familiar with this. But because it's on the X chromosome, this means it's going to be an X-linked disease, and all X-linked diseases, all the high-yield X-linked diseases, with the exception of one we'll talk about, are X-linked recessive. Males only have one copy of this gene, so males are usually the people affected by the symptoms of this disease. Okay? Worldwide, it's one of the most common single gene disorders. So there's a huge amount of people who have this disorder. Estimated 400 million worldwide. That is a huge number for a genetic disease. Huge number. I mean, just incredible number. There's only what's the latest count for the population of the world? 7 billion or something like that, 8 billion, somewhere in there. So if you figure that out, then you'll see it's like 1 in 20, 1 in 25 people or something like that if you look worldwide. So in some areas of the world, the gene frequency is up to 25%. So you get 25% of the males in that population are affected. And the populations with the highest incidence are among those of African, Mediterranean, and Asian descent. Okay? Just so happens that if you actually map the areas of the world that are most affected by malaria, you will see a big overlap of the places where you have the highest incidence of G6PD deficiency. So actually, anytime you see a genetic disease, in particular one that may have relatively severe symptoms, but it persists at a very high level in a population, you need to start thinking there may be something called heterozygote advantage, where carriers or possibly even the people affected by the disease may have some advantage over those who don't. And in this case, just like with sickle cell, which I think you've already seen, sickle cell anemia, I don't know if you've talked about thalassemias at all. Have you talked about thalassemias? Oh, you have. Awesome. So both of those confer, if you're a carrier for those, some protection against malaria. Same thing with glucose 6-phosphate dehydrogenase deficiency. Okay. Now, in America, the incidence is about 11% amongst American blacks. So, and this is one of those things where, you know, when you're doing a patient history, it can be helpful to find out their heritage. Because if you have a patient of African descent or Mediterranean descent, you may, in terms of likely diagnoses, when you see a problem, this may be something you might be more likely to look at, for example. So, what is the actual symptoms of this condition? This disorder manifests almost exclusively in red blood cells. So the only symptoms we're going to see are in red blood cells, and I'll explain why that is in a second. And what happens is we get an episodic hemolytic anemia. Okay, most of the other diseases we've seen are chronic hemolytic anemias, the other hemolytic anemias we've seen. Sickle cell, you could make an argument that it might be episodic. There's certain things that will certainly make it worse and precipitate an episode. So you could say sickle cell would be episodic as well. But this is episodic. So it doesn't mean that it's, so what it means is there is episodes. So it's not going on all of the time. So it's a hemolytic anemia, that's the issue, but it's also episodic. And that's going to be important for, you know, you're potentially trying to diagnose, okay? So it's not all the time, not going on all the time. And so we're going to talk about the things that will actually precipitate the hemolytic anemia. So why does it exist only, or why do you only see symptoms in red blood cells? Okay, there are, in other cells, there's other ways of generating NADPH. Well, first of all, what's going to happen if we have a defect in the HMP pathway? Okay, so HMP pathway does two things. It generates NADPH, it generates ribose for nucleotides, right? In red blood cells, we don't really, there should be enough nucleotides to last in there, so really the compromising the ability to make NADPH is going to be the issue in red blood cells, okay? In other cells, you actually have different ways of making NADPH. Okay, so if you have a defect in this enzyme, it's not that big a deal. The other thing is, as we're going to see, most of the mutations that cause G6PD deficiency, they're mutations that actually affect the stability of the enzyme. That means the enzyme is actually perfectly normal in terms of its catalytic ability. Km, the Vmax, those aren't affected. There's some kind of change in there that affects the stability of the enzyme, so it gets turned over more rapidly. In most cells, that's not a big deal, right? You'll just make more. If you run out, you just make more. But in red blood cells, once they're mature, they've lost that nucleus, they don't have the ability to make more, okay? So over time, if that enzyme becomes defective, then the rest of the life cycle of that red blood cell, it's going to be deficient with that NADPH. That compromises its ability to neutralize reactive oxygen species, and that's what happens here. So it compromises its ability to neutralize reactive oxygen species. Therefore, anything that increases reactive oxygen species is going to cause the hemolytic anemia. And in this case, just like with other diseases, where something goes wrong in the red blood cell, and it looks like it's damaged. Once you start damaging membranes, for instance, the system, the reticulocyte system that exists in your body to turn over red blood cells recognizes, okay, there's damage there, we're removing them from circulation, and therefore you get the hemolytic anemia. Okay, so does that make sense to you why it happens in red blood cells, but you don't really see symptoms in other tissues? Okay, so in other tissues, that enzyme can just simply, if you lose the enzyme after a couple days, you just make more, not a big deal. Can't do that in red blood cells. So as the red blood cells age, and remember, red blood cells last, 110 to 120 days. So all the enzymes that are needed need to last that long, and if they don't, severe things are going to happen. Okay, so people with G6PD deficiency are usually only symptomatic when experiencing an oxidative stress. So under normal circumstances, the G6PD deficiency is not a big enough deal to cause a significant hemolytic anemia where you're going to see some kind of macroscopic symptoms. But anything that increases the amount of oxidative stress can precipitate an episode of a hemolytic anemia and could potentially kill the patient if it's severe enough. So what are some of the things that will precipitate episodes? One is an infection. And you just saw that during an infection, your body itself will generate free radicals, right, to try to fight the infection. But infections themselves will generate free radicals. Microorganisms can generate free radicals. So if you have a patient with G6PD deficiency, you have to be very, very careful if they get some kind of infection, okay? So in an average individual, if they get some kind of infection, you might just say, hey, your body will fight it off, just, rest for a couple days. But you have to be real aggressive with these patients in treating infections, because whenever they get some type of infection, they can now be prone to getting a hemolytic anemia, which could be potentially life-threatening. There are also drugs that produce an oxidative stress, and you will see this later on. There's lists of drugs that patients with G6PD deficiency can't take, or at the very least, you have to be very careful about when giving them these particular drugs, because these drugs and the way they're actually metabolized will actually cause an oxidative stress. In a normal individual, it's not a big deal. In a G6PD deficient patient, it can be. And oddly enough, there are some versions of G6PD deficiency where fava beans can actually create enough of an oxidative stress where you will actually see the hemolytic anemia and symptoms. And fava beans are a staple in a Mediterranean diet, and you also see a high incidence of G6PD deficiency in Mediterranean regions. Why? And you'll also see this term favism, which is a G6PD deficiency. And I haven't really seen a particularly good explanation yet why, because I'm sure you're asking why is it only certain variants. When I say variants, I mean there's different mutation types that cause G6PD deficiency. I haven't seen a good explanation why it affects some mutation types and not others. But it's one of those things that remains to be proven. It may have been proven, and I just don't know what the explanation is, but it's something you will certainly encounter. So if you have a patient who's deficient in G6PD and they undergo some type of hemolytic anemia and you catch it, if you can remove whatever that oxidative stress is, like you can treat the infection and try to make them recover quicker, or whatever else is, if it's a drug, you remove the drug. The patient will recover as new red blood cells are being made, okay? And if they can avoid another oxidative stress, they're going to be fine. Okay, so G6PD deficiency, and I guess I didn't write it there, but you remember we talked about pyruvate kinase deficiency, right? Pyruvate kinase deficiency, that was in our glycolytic pathway. I told you that was the second most common enzyme deficiency that causes a hemolytic anemia. Now, there are other hemoglobin defects that cause hemolytic anemias, but we're not counting, hemoglobin isn't an enzyme. Anyway, this is the most common enzyme deficiency that causes a hemolytic anemia. So one of the things when you're doing a differential, when you've got a hemolytic anemia, is you would be looking at both possibly pyruvate kinase deficiency and G6PD deficiency, as well as some other things as well. Certainly, the fact that this is episodic could be a hint that it's G6PD deficiency. But one of the diagnostic tools that can be used to differentiate this from other hemolytic anemias is in G6PD deficiency, you'll see these Heinz bodies. And Heinz bodies are precipitated hemoglobin. There are some other things that cause Heinz bodies, not just G6PD deficiency, but certainly this is the most common thing that causes it. But it's the only hemolytic anemia that you're going to see Heinz bodies in. So it's precipitated hemoglobin. So what happened? During an episode, you couldn't neutralize reactive oxygen species. What are red blood cells full of in terms of protein? Hemoglobin, right? So those reactive oxygen species react with hemoglobin, and when they do, it can actually cause hemoglobin to precipitate out, and you can actually observe it under the microscope. And that's what these Heinz bodies actually are. Okay? All right, so that's the story of the HMG pathway. And now we're going to spend a little bit of time, just last 10 or 15 minutes here, talking about ethanol metabolism. Everybody's favorite metabolic pathway, right? Especially after blocks. So ethanol, I'm sure none of you guys drink. I'm just kidding. Ethanol itself is a unique chemical. Ethanol is really different from, so we've talked about polar and non-polar and fat-soluble and water-soluble. Ethanol is actually both. Ethanol is very water-soluble, so it can exist in water very good. That means it gets in your bloodstream just fine. It's also, unlike other water-soluble things, it can very easily cross a membrane. So it gets inside cells very easily, okay? That's part of its wonderful effects it has on the human body. So it's very easily absorbed and readily enters the bloodstream, and then readily enters all different types of cells. Now, a small amount, if you're ingesting normally through the mouth, a small amount will be metabolized by cells in the upper GI tract. So you will get some metabolism in your stomach, for example, and a small amount will be lost through the lungs. So you'll breathe a little bit out, and some will end up in the urine as well. These are very small amounts. So you can get rid of ethanol in those manners, but the majority of the ethanol is actually going to be processed by the liver. Okay, so the liver is going to be the big organ that's going to process ethanol. there's two different systems that actually process ethanol. There's two different systems that perform the very first step. So actually metabolizing ethanol only has two enzymes. Okay? There's only two enzymes in that pathway. And what we end up with is acetate. And once we have acetate, we can actually shuttle that. You can actually add CoA to it. Now you've got acetyl-CoA. So you already know what we can do with that, right? We can shuttle that into whatever we need to. So the two different systems that deal with ethanol in the beginning are alcohol dehydrogenase, which is the enzyme we're going to focus in on today. There's also a microsomal ethanol oxidizing system, this MEOS system, that actually uses cytochrome P450 enzymes. Therefore, it actually uses NADPH, which we saw in the HMP pathway. We're not really going to talk about that system today. It doesn't process a lot of the alcohol. Now, if you have somebody who is a heavy drinker or an alcoholic who drinks a lot, this microsomal ethanol oxidizing system gets induced and processes more of the ethanol. But in a normal individual, this is going to be a relatively small amount. Let's say 10% of the ethanol would be processed through that system. But whether or not you use alcohol dehydrogenase or the cytochrome P450 system, this MEOS system, in both cases you process ethanol to acetaldehyde. Okay? So that's this first step. And it is important in this case, in most of the pathways we talked about, we didn't try to learn all the intermediates. This is a very small pathway, and the intermediates happen to be important, so we do need to know ethanol to acetaldehyde to acetate. Okay? So, in order to actually metabolize ethanol, you really only have to go through two steps. First is alcohol dehydrogenase. So you have the MEOS system, and we're going to leave that alone for now. You have your alcohol dehydrogenase, converts ethanol to acetaldehyde, and then you have a second step which converts acetaldehyde to acetate. Once we get to acetate, that's an innocuous molecule. We can deal with that. We can shuttle it into things you've already seen before. Okay? So this system is going to account for the majority of the processing of ethanol. Okay, so this intermediate in the processing of ethanol, this acetaldehyde, acetaldehyde is very, very toxic. It's a very reactive compound. It's almost like a reactive oxygen species in that regard. Okay, it's non-specific. It reacts with just about anything it can come in contact with. So it'll react with proteins. It'll react with lipids and membranes. So it'll damage things non-specifically, just like reactive oxygen species do. So the toxic effects you see from drinking alcohol, in really what you see from people who abuse alcohol long-term, are because of the toxic effects of acetaldehyde. And I just told you acetaldehyde, or the pathway to process ethanol is present mainly in the liver. It does the majority of the processing. So the acetaldehyde that's actually generated is generated also in the liver. So the big thing you see is liver damage. Okay? And you already know, at least you're familiar with some of the functions of the liver. So those functions start to become compromised when you start damaging the liver. You start damaging it enough, you get cirrhosis, and eventually this will kill you if you don't do something about it. So the buildup of acetaldehyde, so in a normal individual, they process, when they go through this process, the acetaldehyde buildup is not normally enough to see overt symptoms. Okay, this destroying of the liver takes place slowly over time. But if you have a very slow variant of aldehyde dehydrogenase, which is the second step, which means acetaldehyde will build up, or you have a very fast alcohol dehydrogenase, so you get to that, you also start building up acetaldehyde faster than the next step can process it, or a very fast meiosis system where you build up acetaldehyde. People who have those variants of the enzyme can't drink very much because they start to get symptoms of acetaldehyde sort of poisoning, if you will. And that includes you get flushing of the face, you start getting nauseous, even after only a small amount of alcohol. Okay? It's known that it's a common allele in those of Asian descent. Okay? So, and these people can't drink very much. Maybe even a single drink would be enough to make them very, very ill and get very, very flush, very nauseated. Okay? The benefit of having that is those people, when they've done population studies, have found complete protection from alcoholism. Alcoholism does not exist in people who have those variants of the enzyme. And that makes sense, right? If you drink a little bit, so they'll never drink enough to become an alcoholic or to get to the stage where they're actually addicted. There's also a drug called Antabuse. That's its trade name, disulfiram. I just call it Antabuse. It's easier for me to say. But you should know both names. Disulfiram is probably the one you would encounter on a board exam because Antabuse is a trade name. But anyway, you may encounter either one of those. This drug inhibits aldehyde dehydrogenase. Okay, and I want you to know this drug inhibits aldehyde dehydrogenase. And don't make the mistake. I always ask people, okay, what does Antabuse inhibit? And they'll say alcohol dehydrogenase. No. What's going to happen if you inhibit alcohol dehydrogenase and you drink? You're going to be drunk for a long time, right? So you're going to have to eventually breathe out the alcohol. So, that would be a true cheap date, right? You could be drunk for a week from drinking very little. But if you inhibit that second step, that aldehyde dehydrogenase, what's going to build up? Acetaldehyde, and it's going to make you very ill. So this is a drug that's used sometimes to treat people who abuse alcohol or people who are alcoholics. It's used as a deterrent, okay? Because if they drink a little bit, they will get ill. The problem in this case is if they go ahead and decide to drink anyway, what's going to happen? They're going to build up acetaldehyde, so it can actually hasten the liver damage caused by the buildup of acetaldehyde. Okay? And I have seen people who still have drank on Antabuse, and it causes some really bad damage. So what we're left with at the end is acetate, and it can be shuttled into pathways you've already seen. Okay, so I want to look at how alcohol processing is going to affect metabolism. I have to go kind of quick here. We've already talked about what acetaldehyde does, but the processing of ethanol produces large amounts of NADH. I didn't explicitly say that before, but if both alcohol dehydrogenase and aldehyde dehydrogenase both produce NADH. Okay? Now, I've given a little aside there. What I'm going to talk to you about is only if you drink a lot of alcohol, like you binge drink or you drink a lot all the time, or perhaps if you drink a bunch on an empty stomach, which we'll talk about shortly. So this buildup of NADH, and I'm talking about a massive buildup here, so we're talking about drinking a lot, will start affecting pathways. These are pathways you've already seen. So let's just assume we drink a whole bunch of alcohol and we're in the well-fed state. Okay, and so your body will process that ethanol first. Okay, it's going to generate a whole bunch of NADH, right? And so you're in the well-fed state, glycolysis is going on, right? The buildup of that NADH is actually going to shut down. It's going so fast, or your buildup so much, it's going to shut down TCA, going to shut down electron transport chain. So now that pyruvate from glycolysis, where is it going to go? It's going to get shuttled where? In order to regenerate that NAD+, it's going to have to get shuttled into the formation of lactate. So you'll get a lactic acidosis. Okay? So if you're in the well-fed state, you're going to get this lactic acidosis. A secondary effect of lactic acidosis, so in alcoholics, this is one of the things you'll see, a chronic lactic acidosis, regardless of if whether in the well-fed or fasting state. You typically also see gout, which is hyperuricemia, or gout is caused by hyperuricemia. Anytime you have somebody who's at a chronic lactic acidosis, you get a secondary hyperuricemia. Now, if we're in the fasting state, what are you supposed to do in the fasting state? Gluconeogenesis, right? The problem is the buildup of NADH will actually shuttle the gluconeogenic precursors, which could be pyruvate, or it could be oxaloacetate. If you look on the other slide, it'll actually favor the conversion of pyruvate to lactate or oxaloacetate back to malate, which was from the TCA cycle, so you can't perform gluconeogenesis. Therefore, what's going to happen? Well, you're also going to get the lactic acidosis, but now you're actually going to get a hypoglycemia because you can't perform gluconeogenesis. And this effect you can actually see in somebody who drinks even a relatively small amount, but does it on an empty stomach. Okay, as long as you have glycogen available, you're okay because you can actually, gluconeogenesis doesn't have to kick in right away. You can actually maintain your blood sugar from glycogen. But let's say you haven't eaten in 12 or 24 hours, and you decide, to tie one on. In that case, your glycogen stores have been depleted. You start drinking, you start producing a whole bunch of NADH, and you inhibit your gluconeogenic pathway, and therefore you get hypoglycemia. So what's the lesson here? If you're going to go out drinking, you got to carb load first, right? You got to build up your glycogen reserves. Okay. So there's something useful you learned today. Now, we didn't have time to go into it, but ethanol, as you might imagine, affects metabolism of, because of that buildup of NADH, you end up affecting all kinds of metabolic pathways, which we didn't have time to talk about. Right. All right, guys. I think you're not going to see me for a couple weeks now. We're going to till genetics. Genetics is worth waiting for, though. Good stuff. Is there a question? All right, I'll hang around if there's any questions.